All right, so today I'm going to be going over last Wednesday's exit ticket on the cell cycle and mitosis, which had you do a discussion post. So, for a little background, mitosis is the process of replicating the genetic material of the parent cell and distributing it to two new cells, which are often called daughter cells. And a crucial step in this process is the breaking down of the nuclear membrane. So this mitosis can only occur in eukaryotes, as prokaryotes don't have a nuclear membrane surrounding their DNA. And the main purpose of mitosis is to accomplish cell regeneration, cell replacement, and overall growth among the organisms. And what's very important is that these cells will be the same and they will have the same number of chromosomes and genetic information, which is very important. So we can start off by breaking down the phases. The first phase is known as interphase. Technically, this is preparation for mitosis. It's not actually part of the mitosis duplication process, but during interphase, this is when the chromosomes of the parent cell duplicate and become two identical chromatids, and stages such as the G1, S, and G2 stages also occur. And as soon as mitosis ends, the cycle begins right again with interphase as well. It's a continuous cycle. So our next step is prophase, which is when the genetic fibers within the nucleus, known as chromatin, condense into chromosomes, and then the spindle fibers form as well. And then during the metaphase, those fibers will attach to the chromosomes, and because the fibers are centralized on the opposite sides of the cell, it'll cause the chromosomes to line up at the equator of the cell. And then during anaphase, the fibers shorten and they pull the chromosomes apart at their centromeres where they're connected. They're pulled apart to the opposite sides of the cell. And then telophase, which is our final phase, is where the nuclear membrane reforms around both sets of chromosomes that are split up, and the fibers will break down, and then this is where cytokinesis occurs, which is where the cell membrane almost pinches and then splits into two identical daughter cells, which are identical as the parent. So with that background info, we can go ahead and proceed with filling in which phase goes with what over here. So where the nucleolus disappears, this is going to be the prophase. There's actually a phase between pro and metaphase known as the prometaphase, and it can be known as the late prophase, early metaphase. This is kind of where the nuclear membrane breaks down mainly. So if you put metaphase here, I won't count it against you. So then for step 2, where the sister chromatids are lined up at the center of the cell, this is the metaphase. And then number 3, the DNA of the cell is replicated. So this will happen at the interphase, the very beginning. For 4, the chromosomes coil and thicken, becoming visible structures. This will happen at the prophase, where they condense into chromosomes. And cytokinesis occurs at the very end in the telophase. And then for 6, the nuclear membrane fragments. So this is kind of one of those ones that could be prophase / metaphase, but I'm just going to put prophase here for now. 7, the cell doubles the number of organelles. So this is going to be the interphase where it's almost preparing for the mitosis, getting all the components together and stuff, because it does need to split into 2 cells, so the cell needs to make sure it has enough to do that. Excuse me. And then 8, the mitotic spindle begins to form. So this is what happens at prophase, which is then attached to the chromosomes. 9, the nuclear membrane begins to reform. So when it reforms, this is at the very end, which is telophase, where the cells are finally becoming separate into 2 cells. And then 10, the cell is not actually dividing, but carrying out normal activities. Because interphase is not technically part of mitosis, it's not dividing yet, so that would fit there. All right, and then 11, the nucleolus reforms, so this will be telophase. 12 is the centrosomes begin to move to opposite sides of the cell. I was describing this earlier. This is also the prophase. And then lastly, 13, chromatids are pulled to the opposite side of the cell. And this is what happens during the anaphase, where the chromosomes are broken apart and then the chromatids are separated. So yeah, if you have any other questions regarding this, feel free to email me at this email, and I'll be more than happy to help you. Thank you.